Today's topic is Methods of Biotechnology in Principle and Process of Biotechnology, Unit 8, Chapter 4. So, we have seen the development of biotechnology in the previous class, and today we are going to see the methods of biotechnology. In that, the first method is fermentation. So, the fermentation is derived from a Latin word fervere, that means to boil. And the fermentation is a metabolic process. Okay, it is a metabolic process where the organic molecules like glucose, that is the carbohydrates, that is especially the glucose, is converted to acid, gases or alcohol in the absence of oxygen or any electronic transport chain. So the fermentation, it is a metabolic process where the carbohydrate like glucose is converted to acid or alcohol or gases in the absence of oxygen or any electron transport chain. That process is called fermentation. And the word zymology. The zymology is the science that deals with the study of fermentation process, and it is organized in 1856 by Louis Pasteur, a French chemist who demonstrated the fermentation in yeast, that is the fermentation caused by yeast. The yeast has a special enzyme called zymose which converts the which initiates the fermentation process. So because of the zymase enzyme, the fermentation, the study of fermentation is called zymology. So the word zymology is derived, that is arised from the zymase enzyme. So it is called zymology. They may ask in 1 mark also. And there are certain organisms involved in fermentation are bacteria and fungi, so which they live in the absence of oxygen. So oxygen free environment they can able to live. So the bacteria and fungi live in the absence of oxygen, are associated in this fermentation process. They only going to convert the carbohydrates to acids or alcohol or gases. So the bacteria and fungi. Fermentation are helpful in food and beverages industries. So the sugar is converted into ethanol. That leads to alcoholic beverages, the wine, beer. So these and all, the sugar is converted to ethanol, which leads to the alcoholic beverages. Okay, beverages, and the release of carbon dioxide by yeast used in the leavening of bread. So the bread, you have to have some of the fermenting substance which creates the leveling of the bread. A duff like appearance should be there in the bread. So that work is done by the release of carbon dioxide, that is the release of carbon dioxide by yeast. That carbon dioxide is used for the leveling of bread. Production of organic acids to preserve and flavor the vegetables and dairy products. So the organic acids like acetic acid, it is used to preserve, and the vinegar, it is acting as in the vinegar is nothing but the acetic acid. So it is also for flavoring the like some of the acids are involved here. And the dairy products, the lactic acid which converts the curd, cheese, so that and all they are using. So fermentation plays an important role in food and beverage industries. For alcoholic beverage preparation, the sugar is converted into ethanol. The release of carbon dioxide by yeast, it is used for the leavening of the bread, and production of acids is used to preserve and flavor the vegetables and in dairy products too. So bioreactor or fermenter. So for fermentation we need a vessel. Okay. So this is a reactor, that is it is a vessel. Okay. It is a vessel. It contains a mortar. Okay. So motor, a pressure gauge, pH probe, temperature, spranger, stream, the steam pipe here, cooling jacket, oxygen concentration, sterile nutrient medium, impeller, this is the impeller inside it, and nutrient or inoculant, that is it was passed through it. So this is a bioreactor. And now we will see the bioreactor, okay, in detail. So bioreactor is nothing but a fermenter. So it is a vessel. So bioreactor is a vessel or a container, okay, used, that is it is specially designed for the fermentation process. So this vessel, that is this container, is specially designed for fermentation process, okay. So it provides an optimum environment in which the microorganisms or their enzymes interact with the substrate to produce a required product. So you can see here in this vessel there is an optimum temperature and optimum pH. Okay, optimum temperature and pH was provided in the vessel. For what purpose? So they have to interact, that is the enzymes and the microorganisms has to interact on the substrate. So what substrate is used to going to ferment and to produce? So for the product. So it has to, that is the substrate, with the substrate the microorganisms and the enzymes has to interact. After interaction of substrate with microorganisms and the enzymes, the microorganism only produces the enzymes. So the substrate and the enzymes are interacting and produces the product. For this, they need an optimum temperature, that is the pH. And the optimum temperature you have to provide that. Here they named it as optimum environment. In the bioreactor, aeration, agitation, temperature and pH are controlled. So the aeration is air space and agitation means shaking. During the process the reactor will be, when we put a motor, that will shake. That creates the agitation in the reactor. So during the fermentation process you have to give an optimum temperature. So the temperature is adjusted and the pH is also adjusted according to the substrate and the enzymes. Okay, if the temperature and the pH is over more, the enzymes will not work and it will denatured. So the enzymes are sensitive to the temperature and the pH. So because of it should be controlled. That is the things and the agitation, aeration, temperature and pH in a bioreactor has to be controlled. Okay. And the fermentation involves in two processes, upstream process and downstream process. So we will see the upstream process. All the process before starting of fermenter, such as sterilization of the fermenter, preparation and sterilization of culture medium, growth of the suitable inoculum. So these are the upstream process. So sterilization, usually in our home, that is our mom will clean the vessels before using it for cooking. So that is the cleaning process is called the sterilization process. Why? To get rid of the microorganisms, other unwanted microorganisms to be grown in the fermenter, that is in the fermenter. For this, first they have to sterilize, that is clean the bioreactor vessel or fermenter vessel. And next step is preparation and sterilization of the culture medium. So the culture medium you have to prepare on which the organism going to grow. So that medium has to be prepared first, and after preparation is over, the medium has to be sterilized. That the medium is also to be sterilized, means also to be cleaned with the help of by heating, or by the heating is the traditional procedure to get off the microbes, okay, that one, or by using some other chemicals. And after that, the growth of the the particular, in the culture media, the growth, for you have to give a suitable inoculum, that is to initiate the growth of the organism. So suitable inoculum is used. So this is a upstream process, that is the starting process. So upstream means starting. Before starting, you have to sterilize and prepare and have to growth. Okay. Next, downstream process. All the process after fermentation process is called downstream process. So that is distillation, centrifuging, filtration, solvent extraction. So most of this process involves the purification of the desired product. So downstream process means completion, completing process or ending process. Ending process, that is in fermentation. The first is distillation. So the distillation means it has to be drop by drop. You have to collect the material, that is collect the substance, okay, the product, drop by drop. So by drop it got purification. And centrifuging, it is mixing, it is a mixture to separate the wanted and unwanted thing. So by separating the liquid and the sediment portion, that is called centrifugation. After centrifugation, you are filtering the centrifuged material, that is material, by filtration. After filtration, you are extracting the product. So that product is now, it is fully purified one. So downstream process is a purification process which involves distillation, centrifuging, filtration and solvent extraction. So this downstream is called as ending process. Okay, it is completing. No, the upstream is the starting point. Starting process means downstream is completing. That means ending process. So you understand now? Now, the procedure of fermentation. So, depending upon the type of product, the bioreactor is selected. So, based on the product, it may be a carbohydrate or it is on what basis, that is the product may be acid, the product may be alcohol or the product may be gaseous. So, based on these types of 3 products to be appeared through fermentation, you have to select the suitable bioreactor for fermentation. A suitable substrate in liquid medium is added at a specific temperature, pH, and then diluted. So the suitable substrate, for example, the molasses, that is, it is used for preparation of alcohol, no? The sugarcane molasses. So if you take that is a substrate. Okay, the substrate which going to become a alcohol. Okay, so the molasses, like for example, I am saying this you have studied in your 10th standard in ethanol production. Okay, ethanol manufacture and preparation and manufacture of ethanol, you have studied it. So I have, if you remember it, please keep it in mind. Okay, this one. So it is that is the substrate in liquid medium you are taking. Okay, you are taking, and for that what and all should be there? Suitable temperature should be there, that is particular temperature should be there, pH, and now in the liquid medium, that is in concentrated you are taking with suitable, that is with specific temperature and pH, and now you have to dilute it, the substrate, with the help of water. So to make it a concentrated, to make from a concentrated one to dilute one, you have to add some water. So the organisms, microbes, animal or plant cell, subcellular organelles or enzymes is added to it. That is the substrate where you added, and in a liquid medium you are taking it and you are diluting. After diluting it, you are that is adding the organisms like microbes or animal or plant cell or subcellular organelles or any enzyme is added to it. Then it is incubated at a specific temperature for specified time. So now incubation means you are allowing to grow in that medium for some time. So that is called incubation. So that is in a particular temperature, in a particular time, you are allowing the substrate to grow. Okay, the incubation may either be aerobic or anaerobic. So the growth, that is the incubation means the growth. It may be aerobic or anaerobic. Aerobic means in the presence of oxygen. Anaerobic means absence of oxygen. Okay. The withdrawal of product using downstream process method. So after allowing to incubation, after completing of the incubation, the withdrawal, the product you have get and you are taking taking through the downstream process like distillation, centrifugation, filtration and solvent extraction. Through this process you are taking. So procedure for fermentation, the first one is depending on the product type, you are selecting the bioreactor, and second one, suitable substrate, that too in liquid medium you are taking, and you are giving specific temperature and pH and diluted. And the next one, the organisms are added to it. The organism may be enzyme or animal or plant cell, subcellular and microbes. They are added to that particular substrate containing a vessel. And now you are incubating, that is incubating with a specific temperature for a time, for a limited time with specific temperature you are incubated. After incubation, that is before the incubation is may either takes place in aerobic or aerobic. After this incubation is over, you are withdrawing, because you are getting the product and you are withdrawing the product in downstream process like distillation, centrifugation, filtration and solvent extraction. And now we move on to application of fermentation in industries. So microbial biomass production. So microbial biomass is nothing but the cells. That is microbial cells is nothing but the biomass, the algae, bacteria, yeast and fungi. So these are the living organisms are used in this fermentation production, that is microbial biomass production. So the algae, bacteria, yeast are considered as biomass, are grown, dried and used as a source of complete protein called single cell protein, which serves as a human food or animal food. So biomass production means the microbial cells. The microbial cells is the good source for protein. So the single cell protein, so which going to see in the next class. So single cell protein, they have single cell protein and it plays an important role in human food in space and animal food also. So it is highly nutritious one which is taken from the microorganisms like bacteria, algae, yeast and the fungi. So yeast is a fungi only. Microbial metabolites. So the microbes produce compounds that are very useful to man and animals. These compounds are called metabolites. So the microbes producing some compounds that is very useful to the man and the animals, and they are called metabolites. And these metabolites are categorized into two: primary metabolites and secondary metabolites. Primary metabolites. Metabolites produced for the maintenance of the process of microbes are known as primary metabolites. Example: ethanol, citric acid, lactic acid and acetic acids. So that is the metabolites which is produced for the maintenance of the life process of microorganisms. So microorganisms like yeast, bacteria, which is to be grown in the laboratory. For that they are using this. So ethanol, citric acid, lactic acid and acetic acids are primary metabolites which is helpful for the life of the microorganisms in the laboratories. And secondary metabolites. Secondary metabolites are those which are not required for vital life process of microbes but have the value added nature. This includes antibiotics. So this antibiotics is a substance which is extracted from the microbes, but they are not useful for the growth of the microbes. Okay, such as antibiotics, amphotericin B, Streptomyces nodoseus. It is extracted from Streptomyces nodoseus. Penicillin is extracted from Penicillium cryogenum. Streptomycin is extracted from Streptomyces gigris. Tetracycline is extracted from Streptomycin aerophaciens. So alkaloid, toxic pigments and vitamins are also some of the secondary metabolites. Microbial enzymes. When microbes are cultured, they secrete some enzymes into growth media. So these enzymes are industrially used in detergents, food processing, brewing, pharmaceutical. So pharmaceuticals examples are proteases, amylases, isomerases and lipases. So some of the microbes that grow in the culture medium secrete some enzymes from the growth medium. Okay, they are secrete some enzymes, and these enzymes are used in detergents. Okay, for cleaning purpose, detergents, food processing, that is and bread making, biscuit making, there and all they are using, and brewing industries. That is, brewing is nothing but the production of hot drinks. Pharmaceutical, that you know very well, the production of medicine, tablets, example. What are the enzymes are secreted during secreted in the growth medium, okay, they are proteases, amylases, isomerases and lipase. So these are the microbial enzymes which are secreted in the culture medium, that is in the secreted by the microbes in the culture medium. Sorry, in the culture, they are cultured, not in the medium, they are cultured. Bioconversion, biotransformation or modification of the substrate. The fermenting microbes have the capacity to produce valuable products. So they have the capacity to produce valuable products. Example, conversion of ethanol to acetic acid, isopropanol to acetone, sorbitol to sorbose. So this is the sorbitol to sorbose. This is used in the manufacture of vitamin C, and sterols to steroids. So the conversion, bioconversion or biotransformation or modification takes place in some of the valuable products. The conversion of ethanol to acetic acid is nothing but the vinegar which is used as a preservative. Isopropanol to acetone, sorbitol to sorbose in manufacture of vitamin C, sterols to steroids. So thank you for watching the video. If you like the video, give a thumbs up, share and subscribe to Science Easy Tech channel.